with that said, I think now is probably a good time to begin. So thank you everyone for joining the 10th week of the Bahamas Natural History Conference virtual webinar series. My name is Giselle Dean. I am a Assistant Science Officer at the Bahamas National Trust, as well as the main organizer for this event. We really appreciate the support of you all, especially the presenters, the past 10 weeks. We'll be wrapping up soon, so only 2 more sessions after this are left. But thank you for your support. So just to quickly introduce this session, conservation status, species of concern. Species of concern is a broad term that indicates there's a concern for a species regarding threats and its conservation status, but there's insufficient data to further protect it or list it on an endangered species list or an IUCN Red list. So for the purposes of this session, we have expanded this definition to include species already listed on the IUCN's Red list of threatened species. The Bahamas has 18 critically endangered species, 35 endangered species, 58 species listed as vulnerable, and over 100 endemic species, including plants, arthropods, birds, reptiles, and fish. So as we know, some of these species are economically significant, such as the Nassau grouper and the bonefish, which are very important fisheries resources. And some of them are also pretty environmentally significant, such as the Kirtland's warbler, which spends most of its time in the Bahamas as a critically endangered warbler species, as well as single island endemic plants, which can only be found in small areas of islands, individual islands in the Bahamas. So without further ado, we will first be hearing from Dr. David Phillip on acoustic telemetry, using mobile acoustic telemetry arrays to locate bonefish migration corridors and aggregations. Dr. Phillip.
Okay, good morning, everyone. Whoop. Okay, it says I cannot share screen while the other person is sharing her screen.
Apologies, I will stop sharing. All right, you should be able to do that now.
Can I do that? Continue, hold on. Here we go. Okay, perfect. Okay, good morning, everyone. I hope you can all see that.
Yes, looks good.
Good, good. Okay, and you can hear me okay. So I want to start by saying that any one of the three of us, myself, Liz, or Aaron, could present this talk. We're kind of equally responsible for all of this. Because my summer research is usually in Canada and I'm not allowed up there this year, I was deemed the least busy of everyone, so I was nominated to make the presentation. I know Aaron's out in the field, Liz is in the lab working, and so, I get to do this, which is fun. Let me briefly explain what the Flats Ecology and Conservation Program is. This started out with several independent researchers back in the 1990s getting together and conducting some bonefish research. And then we grew as a group, and we still remain basically a large number of independent research scientists that are associated either with NGOs or universities or both. We work together on an independent basis. But back in the early 2000s, when the Fisheries Conservation Foundation was formed, we went to the Island School and formed a partnership with them to work out of their just developing Cape Eleuthera Institute. A few years later, we also added the Ocean Tracking Network on as these partners to work with guides and lodges and anglers to try to come up with a Bahamas-wide strategy for conserving bonefish and looking at flats ecology. So that's kind of how we got started. What I want to cover today is to briefly review bonefish life history, because it's complex and probably not everyone knows what it really is. Talk about some of our initial spawning migration studies, and then briefly zoom through our whole island telemetry studies where we use the mobile array system, and then talk about, close with post-Dorian and COVID strategies for conservation. Okay, here's the bonefish life cycle. Adults, which mature, both males and females, anywhere from maybe 3 to 6 or 7 years, depending upon locations, etc., and species. They are broadcast spawners, means they produce lots and lots of eggs, maybe upwards of 0.5 to 1 million eggs, very small. They go and spawn in large numbers. They extrude, it's external fertilization. The little eggs float. They hatch in 2 or 3 days out in deeper water. And then they grow up to be, after 2 months, they become leptocephali, right? So they hatch out as little larvae and grow as these leptocephali for up to about 70 days out in deep water. And then at some trigger, they swim in towards shore and metamorphose over about a 2-week period, losing half their body length, and turn into little bonefish. The leptocephali look like little glass eels and actually are very difficult to distinguish between larval moray eels or larval conger eels. But then when they become little bonefish, they settle on the bottom, and then they meld into schools of small, similarly sized mohara, where they stay for months or up to a year until they get too large, then form their own schools, move into sandy flats, and then as they get up to maybe 2 years old or whatever, they then start moving in among the adults and move into mangroves. So this life cycle is quite complex because it essentially is a 2-stage system where pelagically they grow into leptocephali, and then later they move into the shallows, change their diet, their metamorphose, and become juvenile bonefish. And that life history, that 2-stage life history, really presents almost essentially insurmountable challenges to any kind of artificial propagation, through logistical issues of having to raise 2 different stages, but also serious genetic issues if you're trying to raise them for any kind of stocking out. So that needs to be taken into consideration and realized when any planning. Okay, question is, however, when and where do bonefish spawn? 15 years ago, when we started some of this telemetry research, you'd ask guides and anglers and every other biologist where they spawn, and you'd get answers all over the map. But most of them said they spawned in the mangroves, which is definitely not the case, even though were purportedly videos of bonefish spawning in the mangroves. They were just kind of bogus. So we decided that we would try to assess bonefish movements and migrations, and in the Bahamas. And of course, the Bahamas is very complex geographically. It's a huge area, 1,000 miles, unbounded by land. So it's a wide open system, and it has hurricanes, and that presents some research problems. So there were a couple of options that you could look at. Some groups are trying to use anyway, mark and recapture of fish that were externally tagged. So there's spaghetti or Floyd tags that they put, that are put into the fish that you can see, and then they're released and then weighted and recaptured later on, usually by anglers. And that there's some serious limitations to that work. One is just point to point, so you know where you've tagged it and you know where you recapture it. It's often months, maybe sometimes years between those 2 points, and all you have are 2 different spots. And oftentimes they're within a reasonably small distance of each other, even though we know that these fish swim hundreds of miles in a month and thousands of miles in a year. So it leads to some serious wrong conclusions, because recapture rates are usually abysmal, and there's biased sampling. So the fish that are tagged are only tagged usually in large groups in very few areas. And when they're captured, they're only captured by anglers where they fish. So there's no real standardized method of sampling. And so really the conclusions that are based on this are not terribly valid at all. There's another method of telemetry where you use receivers and transmitters to track individual movements. Now it's limited by the numbers of receivers. So lots of receivers in a small area gives you good coverage of that area, and lots of transmitters gives you more data, but they're expensive. Transmitters are, you know, $300 plus a piece. The receivers are $2,000 plus a piece. So you can see that a big array with a bunch of transmitters is a major expense, but sometimes that needs to be done. And weather, hurricanes, the receivers are put out in shallow waters, and so leaving them out during hurricanes is a financial disaster. So they need to be removed during periods of storms, et cetera, which presents a logistic issue, but not insurmountable at all. Okay, so what we have chosen to do then is to use kind of a mobile array approach, where we'll ask a research question in an area, put an array out, put transmitters out, put that out in October, November, when the spawning season occurs through May, and then pull those receivers, download the data, et cetera, and hold them in safe places on the ground during hurricanes. And then we can move them and adjust the array every year. So I want to quickly go through the original spawning migration study we started back in 2006. It was run out of the Cape Eleuthera Institute. It was really spearheaded by Andy and Sasha Danilchuk and Karen Murchie down there. And you know, this was back when we really didn't know where they might spawn at all. Here is a closer view of things. Rock Sound is up in the upper right-hand corner. That's Rock Sound there. The airport is up very north. And this is Powell Point. The Cape Eleuthera Institute is located out here on the point. Okay, and in this photograph, it shows where each receiver is. Each one of these white dots represents a receiver, and they stretch from Starved Creek around Powell Point down here off the drop-off. This is Exuma Sound up to the west, and the deep water comes extremely close to shoreline, sometimes only 200 meters from shoreline. Okay, so each one of these white dots is a receiver, and they are covering the shorelines, flats areas, and areas adjacent to deep water. As fish would, they're listening for fish with transmitters. Okay, and so this is what the receivers look like. You can see on the right, it is just strapped onto a rebar that has been cemented into a block. And this is over here on the left is our high-tech launching system, where we just hand Aaron a cinder block and he goes screaming down to the bottom and make sure it stands upright. Okay, and we pick them up essentially the same way to download. And you need to actually pick them up to get the data off of them. So they just sit down there and are constantly listening. For transmitters, we actually implant them surgically. So we anesthetize them in clove oil. We make a small incision, put the transmitter inside, suture them up, and then we put them in fresh water. And during this process, we have fresh salt water pumping through their gills. And we then revive them in fresh salt water and then release them with the rest of the school. So we obtain these fish usually by seining, and we get a number of them, put transmitters in a few, return them back into the school that is corralled within the seine, and then open up the seine and they all swim out. We find that we get much better survival that way than if they're released independently. And we find that seining is much better than angling as far as survival goes. And I know some groups don't use anesthetic and instead just put the head underwater. I think survival is much poorer that way, and I'm not sure that how animal care procedures at universities would ever allow that, but some are still going on. Okay, so here, this is the array again, and we put receivers, 100 receivers, essentially around this point, all the way through these tidal creeks that are here, a big, huge poison flats, and in past Rock Sound, into Rock Sound, airport flats, etc. So they were stretched all over there, and then we waited and listened. And that we put them out in late October and listened for 2 to 2.5 months, and then we went out and downloaded the data to see what we found. Many of them would kind of migrate around for days at one location, and then move to a different tidal creek and move around for days in another tidal creek. But what we did notice was that periodically on the full and new moons, a bunch of them did something very different. And they all just took off. Those that went migrated large distances in one day, up to 10 or 15 miles together, all the way around to this one spot around the point near deep water. And let's look at that point. This is a close-up of Powell Point. Here, right, is the where my cursor is. I hope you can see my cursor. That's the island school, and the Cape Eleuthera Institute is right next door. Where they were going was this little no-name harbor. This whole area used to be a resort with a golf course and everything else that went under in the late 70s and has gone feral. During that period in the 70s, they converted a natural blue hole here by dredging out and made this harbor here for home sites that were never built, but it's still a remnant of the blue hole and this area. And that's where they were going. We found that each of these new moons had some tagged fish driving into or swimming into that area. So we waited until the next moon, got a manual tracker that you could move around on the boat and listen for things, and we went and we waited until it was actually a full moon, and we went swimming to see what was in there. And this is what we found: was a huge ball of bonefish in about 15 feet of water in that protected area back in there, just slowly rotating around. Extremely dense, not doing anything that you would see out on the flats. It was just this massive aggregation. So we watched that, and then in the afternoons, many of the fish started going up to the surface and gulping air, something that you hardly ever see a bonefish normally doing. Then as the sun, as it was setting and almost hit the horizon, this whole massive school started moving out of the mouth of No Name Harbor here. Here's Karen laying on the boat in the middle of the night with the listening, and we were tracking the fish movements there. And these fish moved out at 7 o'clock just as the sun was setting and started moving on down here. High tide was at 8 o'clock. Okay, and they moved down here, and at 9 o'clock, they shot offshore. They shot offshore and went spawning out here, we thought, and then they would move down. They came back in after 2 hours, and then slowly made their way back in there before the sun came up. High tide was at 8 o'clock. They were out off on the deep wall from 9 to 11, and we saw that, in fact, they did this 4 nights in a row, and then they all left No Name Harbor and swam back to the flats from whence they came. So we believe that was spawning, and they are laying their eggs off the deep water as tide was going out to carry them out into Exuma Sound to avoid predation by smaller fish. And since then, we've looked at a lot of these spawning aggregations. The migration out always occurs at night, probably for protection against predators. And we've tracked them actually and watched their behaviors by putting light sticks on some individual fish, and you can follow the school with those light sticks and see what they're doing. And so they are spawning off the wall where it gets close to the shoreline for protection. Okay, well, this was some important information, and we wanted to get this out to everyone. So what we did was we organized the first of a series of Bahamas bonefish conferences. And we shared these results with the fishing community, discussed concerns and issues, and then we worked with them to develop a research agenda, because clearly fish aggregations present a serious conservation challenge. One person with a net could pull up a lot of those bonefish in one of those aggregation schools. And so during these bonefish, the first one, we developed conservation strategies, and the main strategy was was to protect spawning aggregations. And so we wanted to identify spawning sites, migration routes, nursery areas, etc. So we knew one site, which was here. But as you can see, I mean, certainly bonefish are located in all the light blue, shallow areas around all of the coastlines. So it's a very complex issue. So how did we figure out a strategy to go about this? So we chose to look at Grand Bahama first. For one reason is the bulk of the research, the bulk of the fishing and bonefish populations are on the north side, the shallow side where all of the flats and mangroves are, but the deep water is all on the south side, and they'd have to get there. You could get there from the west end, or you could go around the east end through the Deepwater Key Cut. They used to be able to go through Hawksbill Creek, but the causeway that was built to build the port there cut that off. Now there is the Grand Lucayan Waterway, but it's a man-made canal, and no one was sure whether fish ever used that. So this was the first year of the study, and Karen Murchie, who was then a professor over at the college, now University of the Bahamas in Grand Bahama, kind of spearheaded this. And this shows the locations of the arrays of the receivers that we had built in our array. And we concentrated on that canal area this first year by putting in 30 bonefish, tags into 30 bonefish, 5 in each of these 6 areas. Then we listened to where they swam, and lo and behold, several went around the west end, several went around the east end through those cuts, and a bunch of them did use the canal, went through and back and forth through there to here. So we have fish going to the other sides, to the deep end, to get to the spawning areas in 3 different ways, and they kind of seem to go to 3 different areas. So then for year 2, we added way more receivers, and in more details around the potential spawning aggregation sites. We tagged lots more bonefish and spread it all out through the northern and southeastern parts of Grand Bahama. And this is what we found. We did find 3, and only 3 spawning aggregation sites, a large one on the east end, a pretty good one in the middle, and then a smaller one out on the part near the free port. And this one may have historically been much better, but it is quite close to an awful lot of disturbance and may be kind of on its way out of existence. So that's a concern. We then moved and did the same experiment. I want to summarize a few different islands now. We did the same experiment. This was Georgie Burris's thesis on Eleuthera. And this shows the receiver locations. We did this for 2 years, and we tagged over 100 fish. And there we go. It shows the receivers and the tags that we did. And this is the array that we have been working on now. This is the northern Cays, from Walker's Key all the way down. We've done all the way down to past Green Turtle Key to Guana Key, and the parts of Little Abaco Island. And so what we were going to do this past year was the rest of Abaco down here, but we got shut out with the hurricane and Dorian. But that's the way we approach this, with receivers and transmitters over usually a 2-year period. And so this is what we've found to date. The red dots are confirmed spawning aggregation sites. The yellow ones are suspected, and we need to confirm those with our next year's worth of spawning data. We have just 3 in Grand Bahama. We've located 3 in the northern Cays. Only 6 in Eleuthera. So there's a big conservation issue. It would be very different if there were 5,000 of these spawning aggregation sites spread throughout the Bahamas, or even 500. But if there really are only 50, which might be the density that we're going to find, then every one of these becomes extraordinarily important for the conservation, of the species. Okay, so here's what we're proposing to do now post-Dorian, and if we can work our way through the COVID issues as well. We certainly want to complete the spawning location IDs around Abaco and some of the southern islands like Moores Island, et cetera, to essentially finish the Little Bahama Bank and find out how many spawning areas there are there. Then we want to hold, and we would like to do that over this winter, hold the Bahamas Bonefish Conference at some point in 2021. Of course, the details of when, where, how that's going to happen are still being discussed among the participants and the organizers because of COVID, the changing COVID landscape. But we're going to update the guides on these new research results, and we're going to try to work with everyone to propose a strategy for protecting these aggregations in the corridors. Certainly aggregations are much smaller than marine protected areas, much smaller than national parks, et cetera. But still probably need to have some level of protection, as well as the corridors, the shorelines at which the fish are migrating to those. We want to work on developing the whole next research agenda, maybe doing this further south, maybe doing some other types of telemetry work, looking at nurseries and migration, not from nurseries to adult foraging areas, et cetera. But critical to all of this would be to implement publication campaigns. Because the local people in the Bahamas need to know that and the government needs to know the issues associated with the rarity of spawning aggregations and how susceptible they are to being destroyed by dredging or any kind of development. So that they need to be built into national plans for developmental activities. Okay. Finally, close. In a attempt to accomplish all of this work, what we've done at FCF is we've formed a new advisory council, and for each of the different projects. And for our Bonefish project, we've assembled over a dozen of the scientists that are doing most of the research all throughout the Bahamas on bonefish and flats, as well as a number of guides and lodge people, lodge owners, anglers, and conservationists that are concerned and dedicated to this. And we're planning a strategy on how to support that research, how to develop fundraising methodologies and get donors for some of this. And we hope to work closely with the BNT on developing our agenda and actually putting our research results into action for protection. Thank you.
Thank you, Dr. Phillip, for that talk. Really interesting stuff about bonefish spawning migrations and aggregations.
Certainly.
So we would like to continue, but first, excuse me. This session is being streamed live on Facebook and will be available. The recording will be available on YouTube at a later date, on the Bahamas National Trust YouTube channel. If you'd like to get in touch with the BNHC team, you can do so by visiting our website, bnhc242.com. And you can also email us at bnhc@bnt.bs. So next we have Dr. Ethan Freed, who is the botanist at the Leon Levy Native Plant Preserve, and he will be speaking with us about red listing in the Bahamas. Dr. Freed.
So, thank you and good morning. I want to thank all of you for listening today about what red listing is, we do it, and then about how what we've accomplished so far. So, red listing. It was started in 1964 by the Internet for the Conservation of Nature, published their first red list of species. And in doing so, they created a codified unit for evaluating a species' conservation status and its biodiversity health, and is a great tool to push for action on biodiversity conservation and environmental policy changes. It's also a resource that's often used by governments, NGOs, natural resource planners, and the business community. So a little bit about the red listing assessment process. Need data to do it. The first two types of data that go to and are the most important are the range and the size. The range is something we will refer to really about how widely spread the species is, and then the occurrence is about how many individuals there are in a population, as well as how much space within that extent that that species takes up. And this a species range quite large, but the population size and space it takes up may not be. So here's an example of a mapping of one of the species. This is Ernodia gigantea. Each point that you see here was observed. And if you go rubber band and put it on, then it forms as the extent. The area occurrence, we map out the smallest mappable unit for area of occurrence is 4 square kilometers. And that may seem sort of arbitrary and large, but it was necessary to pick a unit that was going to be applicable across all the kingdoms. So you're talking about plants or animals or fungi. We also look at things like habitat and ecology, what uses and or trade are occurring, and future threats, conservation occurring or will be occurring. Now, based on that, we will assess species at 1 of 8 levels, ranging from deficient, where we really just don't have any information about that species to be able to understand what its status is, all the way to extinct or extinct in the wild. So we evaluate the species based on 5 criteria, and you look at this picture and say, wow, there's a lot there, but I want you to focus in on these 2, which are the important ones for here in the Bahamas. B is talking about the geographic range, both the extent of occurrence, of occupancy, as well as issues of small population size and decline. And when we certainly are red listing plants, these are the 2 that most often are applicable. So what's been done globally and in the Bahamas in terms of red listing? Globally, there's over 120,000 species that have been red listed, including 45,000 species of plants. Here in the Bahamas, there's been a little over 2,300 species assessed, including 253 species of plants. So the vast majority of what's been assessed here have been animals. So what types of species have been getting red listed here in the Bahamas? First of all, ones in which we have special economic group. You can see Zamia lucifunti. This was studied quite extensively by Dr. Colode out of Florida International University when he was working on his PhD dissertation on Zamias in the Caribbean. Another good example is the Andros rock iguana. This was studied very extensively by Dr. Chuck Knapp of the Shedd Aquarium when he was working on his PhD dissertation. In both cases, a lot of studies, the assessments as well as conservation action plans. Other groups are those that have high economic value, whether we're talking about grouper or lobster or conch. These are ones that, because they've been traditionally fished, there's a lot of information about them in terms on where they're occurring, as well as funding to be able to do those assessments. And lastly, for the Bahamas, we have species that occur here, but they were really assessed on a global or regional level rather than actually what's going on in the Bahamas. So Bahamian plant red listing, and this is mostly what I'm going to talk about since I'm a botanist. When we do that, we want to first talk about the global strategy for plant conservation. This is a subset within the Convention on Biological Diversity, and the strategy outlines 5 goals and 16 targets that countries look to try and achieve to have good plant species conservation. Now, in particular is Target 2, because Target 2 calls for an assessment of all plant species within each country. Now, within the Bahamas, those that have been red listed before are those that had a specialist in that group. And it was a problem because we lacked trained expertise in how to actually do red list assessments so that we could assess large numbers of species. To remedy this situation, we developed a series of red listing workshops. The first one took place in November of 2018. This was hosted by the Bahamas National Trust and the Levy Preserve, and it was supported by Botanical Gardens Conservation International and the Missouri Botanical Gardens, who did the actual training. It was a week long. We had 22 participants, including 13 Bahamians that came from the Bahamas National Trust, the Department of Marine Resources, the BEST Commission, and Forestry. And botanists from around the Caribbean, and while it was taught this, the training is not quick, and all the types of data analysis that you need to do plant assessments is what you need to do for animals or fungi. Is really about learning how and understanding the process itself. Now, the workshop also highlighted to the botanists that we do not spend enough time working and collaborating outside of our countries on regional conservation efforts. So this led to another workshop in Havana, Cuba in spring of 2019 that was focused on Cuba Caribbean species. Now, there I worked with 5 other botanists on Bahama Cuba endemics, identifying 44 species shared only between the Bahamas and Cuba that needed additional work. This in turn led to another workshop in September 2019, hosted by the National Trust and the Levy Preserve again. And this one focused entirely on Bahama Cuba endemics. And there we assessed 26 species and submitted them for review. So very briefly, where do we get all the data when we do this, these assessments? First of all, it comes from personal knowledge of species. It is about getting people together who are experts in these species and the flora of different areas and talking about plants. Now, for some species that are very well known, that we have a lot of experience with, this is relatively easy and gets us going quickly on the assessment. Now, for others, we go to herbarium specimen data. An herbarium specimen is a dried plant collection that was taken from someplace and brought back to an herbarium. On the herbarium specimen, there's the name of the species, the island it was collected on, the settlement, in this case, Hawk's Nest area, and then usually information about the habitat. And then lastly, fieldwork. For those that we don't have either a lot of herbarium specimens of or personal knowledge of, we have to go out in the field, track down those species, record what the populations are, and understand their sizes and status and risks. For me, this is the fun part. Now, we have a number of online tools for doing assessment. In particular, are GeoCat and SIS. GeoCat is a geospatial mapping website where you can go and you can directly input data points for where you know species occur. You can also download information from other websites. This website is also useful in that it will determine both the extent of occurrence and the area of occupancy for that species. Once we have all that, we move over to the Species Information Systems website. This is an IUCN website and allows you to go in and directly input all the data that you have, including your maps and photographs, and then submit it for, to the IUCN for review. So what have we done so far in terms of plants here in the Bahamas? We have a little over 1,400 species of vascular plants that are known here. And as I said, so far 253 of them have been assessed. So this puts us at about 18% of the total for the country. So only about a fifth of the way there. And we have a lot of work left to do. Right now, we're focusing on the 89 endemic species that occur here, and in particular, the 31 single island and the 19 double island endemics, in addition to working on those 44 Bahama Cuba endemic species. So far, since that training, we surveyed, assessed 36 endemic species, had them reviewed and accepted, and 16 of them have currently been published. The other 20 should be coming out in the next publishing cycle once we get past COVID. As you can see, of those 16 that have been published, they're almost all coming out as endangered or critically endangered or threatened or near threatened. Only one of them came out as least concern. And this is unsurprising, is that we focused on those endemics, specifically those that have very restricted ranges. Of those 16 that have been published, 8 of them are Bahamian endemics and another 8 are Bahama Cuba endemics. So I want to talk a little bit about some of the species we've assessed. This is Spermacosi felis insulae, assessed as a critically endangered species. It's also known as the Cat Island false buttonweed. It's a single island endemic only to Cat Island in the Hawk's Nest Marina and Resort area, and a small subpopulation in the Newbite area. It was assessed as critically endangered because the extent of occurrence is just 12 square kilometers, and the area of occupancy is just a little over 24 square kilometers. But in truth, the area of occupancy is even smaller than that. This is the Hawk's Nest Marina right here. This is their runway. And the habitat that occurs in is a very narrow band, not much more than about 30 meters wide, that occurs right along the coast here for about a mile. All of this area is in privately held hands, and our ability to do in situ conservation is essentially zero. And large areas of it are being developed for housing. Being that occurs in just 2 locations and is experiencing a continuous decline in the extent of occupancy, the area of occupancy, the number of subpopulations, certainly the quality and area of habitat, and the number of mature individuals. It's under significant pressure from a lot of different directions. Here's the habitat right here. You can see that it's being invaded by Casuarina equisetifolia. And in the areas where housing has been developed, you also see the invasive Scaevola ticata. It is also just slightly more than a meter above sea level. So as we undergo climate change and sea level rise, this area will continue to get inundated by seawater, and the habitat will decline. It is also directly affected by pollution from humans. Here it is right here, struggling to hold on in the midst of what is a dump right by the Hawk's Nest Marina area. It's thought that this species will continue to decline due to these current threats to the point where this species will likely become extinct. Another example is Cyperus corellii, the Bootle Bay sedge, another critically endangered species. It is another single island endemic known from just 2 small populations on Grand Bahama that are less than 3 kilometers apart, and the entire area is in privately held hands. We have, again, no ability to do any in situ conservation for this species. It's facing a number of threats. The population is, one, quite small. We estimate there's fewer than 250 individuals and fewer than 50 mature individuals between the 2 subpopulations. As you can see, the extent of occurrence is very small, just a little less than 0.2 of a square kilometer, and an area of occupancy that's just 12 square kilometers. But like the spermacosi species on Cat Island, the habitat is actually very narrow. It's a dune species. The habitat's only about 150 feet wide. So the area of occupancy is far less than 12 square kilometers. It faces a number of threats as well. It is being invaded by both the Casuarina equisetifolia and the Scaevola ticata. It also faces pressures from housing development. This is an area where they're building coastal tourism housing. They've come in, they've dug out the entire area and put in a house, and then planted along the dune ridge here more of the invasive Scaevola ticata. There was also a population in the failed West End development that occurred. They came in and did dredging and then took the dredge spoils and put them on the backside of the dune, reducing the habitat in this location to an area that's less than 5 meters wide. Overall, there's been a decline of at least 25% of this species, and it's thought that the species will continue to decline to the point where this will, likely to become extinct in the wild. Another one is Euphorbia longinsulicola. This was assessed as endangered. That's another single island endemic, known only from a few selected areas on the Atlantic side of Long Island. It has quite a small extent of occurrence at 36 square kilometers and a smaller area of occupancy of 32 square kilometers. But again, this is a coastal species. It occurs just along the dune ridge here. Habitat that's quite narrow, not much more than about 10 to 15 meters wide. Now, it does not face the same pressures as the others too. This area of the Long Island coastline was surprisingly free of invasive species, and the vast majority of it had not actually been developed for any housing. But like the other 2 species, being a coastal dune species, it is under direct threat from rising sea levels. So what's next for red listing for the Bahamas? Certainly lots more data that we need, lots more field work to be done to go out and track down all of these species that are data deficient and try and assess their populations and conservation status. We need to continue to partner with within the region so that we can do assessments of species that may be Bahamian-wide but regionally rare, as well as vice versa. And lastly, we need to push and encourage the Bahamas government to include protection for these species and require surveys for red listed species during environmental impact assessments. So as projects do go on, these important and endangered species are surveyed and accounted for. I want to say thank you.
Okay, hi, sorry. Thank you, Dr. Freed, for that talk. Always love seeing endemic species, especially Bahamian endemics. So thank you for that. So continuing with the presentation. First, I just have a brief note to share with you all. The Bahamas has this pretty new field guide called the Natural History of the Bahamas. It's small enough to carry on trips, and it is ultra comprehensive guide of terrestrial and coastal floral and fauna. Dr. Freed has contributed it, as you can see by the authors at the bottom of the screen there. They include species common names and scientific names, locations, and detailed descriptions of the species. It was written by experts in Bahamian ecology with contributions by more than 30 other naturalists and researchers, and covers organisms studied in the Bahamas and the Turks and Caicos Islands with more than 700 color photos. It is for sale in New Providence at Logos Bookstore and Media Enterprises, as well as all national park offices that the BNT manages throughout the Bahamas. The book can also be purchased online at book retailers such as Cornell University Press and Amazon, including digital versions. All royalties from the book sales benefit the Bahamas National Trust, so it's a great way to support BNT as well as this conference. So I have a copy of this book. It's a great resource to have if you're into that kind of thing, which I'm sure many of you are, which is why you're here. So I definitely recommend picking up a copy if you're able to. So lastly, but not leastly, we will have our own avian science officer, Bradley Watson, speaking on the Kirtland's warblers of the Bahamas. Bradley, whenever you're ready.
Good afternoon. How's everybody doing? Can everybody hear me?
Yep, we can hear you. Could you share your screen when you're ready?
I will do so immediately. And I think it's sharing now.
Yep, if you go into full screen just to make sure everything's working okay. Okay, cool. Thank you, Bradley.
Thank you. Good afternoon. So I started out this presentation thinking about Kirtland's warblers specifically, and then I realized that I should use them as a model to consider species of concern, across the spectrum of bird species that live in the Bahamas either part of the year or the entire year. So my name is Bradley Watson. I am a science officer at the Bahamas National Trust, and I specialize in bird matters, in studying the birds of the Bahamas. So why should we be concerned? Or why should we be concerned about particular bird species? The talk is going to go through current concerns, past successes, and then the way forward. How do we capitalize on our past successes to create future successes? So one of the first things that pop out, and the Kirtland's warbler is a great example of that, is that some species have unique life histories, and so the conditions for them to breed and sustain their population size depend on unique factors in their environment. This is a photo of a Kirtland's warbler nest, and this nest is actually on the ground. The tree that it's under is called a jack pine tree. And for these birds to nest, the trees have to be quite young to allow the boughs of those trees to touch the ground and camouflage that nest. This means that we have to continuously create habitat for these birds. This habitat was created by fires in the past, but when fires were reduced by campaigns to reduce the negative effects of these forest fires, we also lost habitat for the Kirtland's warblers. And so we realized that we need to conserve habitat for these birds because they have a unique life history. Another reason to be concerned, and this is another photo of a Kirtland's warbler, is there's threatened populations. And so if you see the numbers of a given species decreasing, it's a motivation for us to take action. And the first action is actually to observe. And so we can see that the surveys of Kirtland's warbler populations began in 1951. Just to highlight a couple of steps on this timeline here, we would note that the first census in 1951 and the first conservation efforts were established in 1957. These efforts included creation of habitat, but it wasn't formalized until the U.S. Forest Service dedicated a tract of land for Kirtland warbler conservation in 1963. And you can note that the populations didn't begin to increase until about 1989. And so it takes a while for these changes to actually become formalized and then for them to actually take effect. And so the sooner we recognize that there are decreases in populations, the better off these species will be. In fact, we shouldn't wait for them to decrease in population. So another reason to be concerned, if you look at this map, you can see that it's a track of Hurricane Dorian. And it shows the Pine Islands of the Bahamas, which are Nassau, Andros, Abaco, and Grand Bahama. And so there are Bahama swallows that are resident in Abaco, Andros, and Grand Bahama. And then we can have even smaller ranges where you find the Bahama warbler, which are only found in Abaco and Grand Bahama. And then the range can decrease even less to the range of the Bahama nuthatch, which is only found in Grand Bahama. And the last time it was seen, it was reported in a very small area of Grand Bahama. So one reason to be concerned is when you see that a species has a restricted range. This makes the species vulnerable to what they call stochastic events, just random acts of chance like hurricanes that can knock out a population. So that's another reason to be concerned. Another reason to be concerned is a transboundary nature. So this graph shows the probability of occupancy of a Kirtland's warbler over the year. And so this was a study done by Nathan Cooper using light level geo trackers. And so you attach a tag to this bird and take the tag back off a year later, and that tag tracks where this bird is, mostly depending on latitude. So moving north and south. So this bird spends half of the year in the Bahamas, half of the year in Michigan. And then you have the path that it takes to move between those two areas. The challenge here is that now you have to protect the bird in two entirely different jurisdictions. And with that challenge comes logistical challenges, financial challenges, even political challenges. Right now, travel between these two countries is at a standstill. And so we have to consider these factors when we think about these species we're trying to save. So then we also think about the cultural significance of these birds. In this image, you have a white-crowned pigeon, and this bird actually does travel between the Bahamas and Florida at times. It also travels between islands, and so while the Bahamas is just one country, we have different almost jurisdictions where things happen differently, and you have to approach the problem, if not from a different angle, at different time points or with different efforts. This bird is culturally significant because there's a deep history of hunting these birds in the Bahamas. And what's remarkable is that the people who know the most about this species are the hunters. And these are the people who feel the deepest connection and are most motivated to conserve that species. And so we have to recognize that, we might be concerned about a species because it's important to us. And that's valid and is relevant. And we should work to conserve these species and keep common birds common. So we're going to talk about some challenged species, some of the species that we want to think about going into the future, species of great concern at this present moment. I call them the pine yard tree because they share a unique habitat, and this habitat itself is of great concern at this point in time. So the first bird is the Bahama swallow. This bird, as I said before, it lives in Andros, Grand Bahama, and Abaco, and it is listed as endangered with the IUCN on the red list. And so there is great motivation to conserve this species. And Maya Wilson, who recently completed her PhD, her dissertation was focused on this species, and she elucidated a lot of the life history that we need to understand in order to have effective conservation efforts. And so this is the Bahama swallow. Another bird we should consider is the Bahama nuthatch. As I noted before, this bird exists only on Grand Bahama and has significantly reduced numbers. And so this, in my opinion, is one of the examples where we're watching a star kind of flicker. And the last time it was seen is in 2018, and it should motivate us to take action as quickly as possible, because this next bird that we're going to talk about is the Bahama warbler. And it inhabits Grand Bahama and Abaco. And we saw great numbers of this bird in 2019 when we visited there in July. But we went back in February of 2020 after Hurricane Dorian, and we didn't see not one of these birds. Now, they weren't singing, they weren't it wasn't breeding season yet, and so that might be the cause. But you know, when you see the pattern of what happened with the Bahama nuthatch, it makes things very concerning for this Bahama warbler. And so the habitat that these birds rely on is, you know, the pine lands. So the pine forests of Abaco, Andros, Grand Bahama, and Nassau, New Providence actually was also a pine island previously. And we can note that, you know, the effects of development are a reduction in natural spaces. But we can have an impact on that by, you know, being concerned and conserving natural spaces. So we're going to talk about the Bahama swallow for a moment. Like I said, Maya Wilson did some very good work on this species, and she recently released her dissertation. This photo was taken during some of the work that they were doing. That's the photo of a camera inserted into the nest of a Bahama swallow in a snag, which is a dead pine tree on Abaco. And so this hole was actually created by a woodpecker. And when we talk about unique life histories, this actually requires the pine tree to be there to die, a woodpecker to create like a nest cavity, nest in there, and then the following year, a Bahama swallow can make use of that cavity. And so that's some of the intricacies we have to consider when we manage these species. To layer on top of that, when we talk about the Bahama swallow, it's one of the pine yard 3 that actually can nest outside of the pine yard, but those nests aren't quite as successful as those created in pine trees. And so you can have a hairy woodpecker, which is found mostly in a pine ecosystem, and they create larger nest cavities in these pine systems compared to a West Indian woodpecker, which might nest in an urban situation in a telephone pole, creating a smaller nest. And Dr. Wilson found actually that nests in telephone poles are smaller, created by West Indian woodpeckers, and less successful than nests found in pine systems created by hairy woodpeckers. So it's a very intricate relationship, and we have to consider multiple species in these systems when we go about conservation efforts. So the Bahama nuthatch is also a cavity nester, and it's restricted to Grand Bahama. And as I said before, it was last seen in 2018. And after Hurricane Dorian, we're very concerned because I'm going to show you a quick video of that habitat after the storm. Excuse me. So this is an area of pine forest in Grand Bahama. You can see that there was a fire afterwards. And this system doesn't offer very many resources for a Bahama nuthatch to persist. And so conditions like these are what make us concerned for these species. So the Bahama warbler, like we said before, was seen very frequently in Grand Bahama previously, but not as frequently now. And we actually did something to kind of recognize this change. And so we have to recognize that our actions of concern, you know, this is a participatory process. Dr. Fried spoke about red listing plants with the IUCN. And so previously the Bahama warbler was listed as near threatened. And we wanted to recognize the damage that Hurricane Dorian inflicted. And so made petitions, collaborated with Dr. Bill Hayes, George Wallace, Dan Levin, Dr. Dan Levin, and Scott Johnson, as well as a few other people to recommend that the IUCN list the Bahama warbler as an endangered species. And so It's a small step towards making some changes, but I think it's a small step in the right direction. So we're just going to talk about a few success stories, a few ways that we have had positive impacts on species of concern. So the Caribbean flamingo nests in Inagua, and we know that in the 1960s, there were aerial flyovers by the Royal Air Force that disturbed these birds' nesting populations. These are anecdotal reports, but since then, the areas have been protected such that planes cannot pass lower than 3,000 feet above these spaces. And this is just one step that was taken. We also created the Inagua National Park to protect these birds as well as other species that inhabit those areas. And so now we have a thriving community, a thriving population of Caribbean flamingo in that park. And what's interesting about these birds is that there's also the transboundary nature that is important, because as our populations were studied and banded by the Audubon Society in the 1960s, they found that birds from the Bahamas were seen in Cuba. Later on, as populations began to increase, we also saw increases in populations in the Dominican Republic and on the Yucatan Peninsula in Mexico. And so we note that, what we do here locally can have a global influence, or at least a regional influence. So we want to note that policy also has a very strong impact in our conservation efforts. And so while it might not seem as exciting as going out and banning flamingos, establishing policies that protect species far into the future are extremely important, especially when we consider that some species are conservation reliant. And so when we have a thriving flamingo population, it doesn't warrant a reduction in our policy or in our protective efforts. And establishing policies like this will secure the future for these birds. So we're going to talk about the Kirtland's warbler one more time. And so we note that there was a really good bounce back for the Kirtland's warbler. And actually they were delisted. They're no longer considered an endangered species, but they are conservation reliant because of their unique life history. And so this picture right here was actually taken in the winter of 2019 on Cat Island, where research is still ongoing, trying to determine what factors influence the success of a Kirtland's warbler on the wintering grounds. The wintering grounds in the Bahamas are extremely important to these birds because that's where they forage in preparation for their migration back to Michigan to breed. So the mass of these birds before leaving for their migration has a direct influence on their success in migration and then breeding in the breeding grounds. So we were looking at birds and trying to compare patterns in the vegetation on Cat Island to the abundance of birds in those areas. And these birds were actually found to live on Cat Island, Eleuthera, San Salvador, and Long Island mostly in the Bahamas in the wintering grounds by research conducted by Bahamians in collaboration with researchers from abroad, namely Dr. Dave Ewart, Joe Wonderly, David Curie, and Dr. Eric Carey, the director of the Bahamas National Trust, instigated this work on the Bahamian side, ensuring that we find answers to these questions. And the method that they used was one of the most sustainable methods possible. It was done by creating capacity. So I know that this is a familiar face to most of you, but we want to note that Scott is the snake conservationist now in the Bahamas, but through work with Kirtland's warblers, like the bird that he's holding in his hand right there, he began his foray into conservation work and actually serves as a mentor for many of us, including myself. So as we build capacity, we ensure conservation efforts into the future for these species of concern. Another example is this photo here. In the background, you see a young Chris Johnson. He's an intern at the Bahamas National Trust who joined us in surveys of Abaco, looking for Bahama warblers and other species after Hurricane Dorian. And Chris is a budding conservationist working along with Dr. Dan Levin from the American Bird Conservancy, as well as our team from the Bahamas National Trust. And I just wanted to point out that building capacity is one way to ensure a future for these species of concern. So just to wrap up, I wanted to go through my kind of framework for how we address these problems. Our threats are oftentimes development and agriculture, but I also wanted to throw in that hurricanes and pandemics are extremely important influences on these populations. And right now, they seem to be top of mind. And so we're looking at Kirtland's warblers as a model species, and we're looking at their habitat in the Bahamas, which is categorized as farms and private land or public land. And so, you know, ways that we can increase the value of this habitat in order to increase the amount of habitat available for these birds in the case of hurricanes and other, you can say, stochastic events that are going to reduce populations. We increase the value of this habitat through ecotourism, recreational activities, and sustainable agriculture. Why do we want to increase the value of habitat? We want to do that because we want to increase the intrinsic motivation of the stakeholders in that community to conserve these species. That's the one way that we will ensure that even after these birds are taken off of the IUCN Red List, or even after, you know, we're not having an active campaign in sustaining a certain facet of this conservation effort, that conservation continues into the future. And so I think I spoke a little bit quickly, and I didn't time myself, but I just want to wrap up. And I did not include my acknowledgement slide here, and that is a mistake. So I do want to acknowledge, namely the Bahamas National Trust, the National Audubon Foundation, the American Bird Conservancy, and numerous other people, Scott Johnson. I'm butchering this, and I'm going to stop there and actually just go ahead and pull that up for you because I don't want to miss anybody out. So I'm going to stop sharing and open up the floor for questions and address the acknowledgements later. I'm sorry, that's a big fumble because I don't want to miss anybody out, but that's the end there.
Thank you, Bradley. I'm sure your collaborators and acknowledgement people will forgive you for that. But great talk. Always important to speak about the protections of and statuses of endemic species. So hopefully we'll see some recovery from the Bahama warbler and Grand Bahama. So that concludes the talks section of this session of BNHCV, week 10, conservation status species of concern. So we hope you can join us for the two remaining sessions of this virtual webinar series. Next week, August 19th, will be deep sea research, and then the following week, August 26th, will focus on terrestrial aspects of the Bahamas. As we, the BNT team, continues to adjust to this new normal, there is still a need for funding events like these weekly sessions. We actually hope to continue these weekly sessions, perhaps not weekly, but have sessions in the future, virtual sessions that will be easily accessible to interested persons. That's still being planned, but fingers crossed. And with your support, we'd love to be able to do that. We would be grateful if you considered donating towards the success of this event and future events. If you have been enjoying the BNHC virtual webinar series over the last 10 weeks, we simply ask that you consider giving us a donation in any amount as a means of showing your support. An email with a donation link will be sent following the session. You can also support us by becoming a member of the Bahamas National Trust if you're not already. Thank you again for supporting us and this event. The floor is now open for questions. Please feel free to submit them via the chat on Facebook or via the Q&A box on Zoom. So we currently have a a couple of questions. So from Facebook, whoops, sorry about that. Dr. Freed, is it possible to collect seeds or offshoots for cultivation to prevent extinction? And has cultivation already been attempted?
Yeah, it is possible. Actually, one of the things I didn't highlight in my talk is how much ex situ conservation work we're doing at the Levy Preserve on Eleuthera. We've managed to now get into our living collection 46 of the 89 endemic species, including 8 of the single or double island endemics. So we're actively working on it. It's just that in many cases the ability to get all that we need to do that work is not always easy or quickly available versus what the threats are.
Yeah, that's completely understandable. Let's see, are there any other questions that haven't been answered in the question box? Dr. Freed, are range maps for endemic plant species accessible on the IUCN or other websites that are available to scientists?
In terms of those that have been published, yeah, that range information is available. But we did a publication in 2014 talking about distributions of the endemic species. And so we did ranges by island, but not specific within an island. And that was sort of some of the work that led to us trying to figure out a little bit more about the populations and do the red listing.
Thank you. I see that some of the questions have been answered via chat, which is fine. Oh, here's a couple more questions. Thank you for the informative talks. I am involved with sawfish conservation in the Bahamas, and I could relate to the identified needs to incorporate species needs into project development assessments, as well as the need for strong protective government policies. Could the presenters share some strategies for engaging the Bahamian government to achieve these objectives? This is from Olga Kobrak. So I think that's open to everyone and anyone.
This is Dave Phillip. I'd say from our experience, the best strategy would be to go directly and first to the Bahamas National Trust and assess their, get their advice on how to proceed and how to integrate it with their plans associated with things like national parks and marine protected areas, et cetera. But I think I look at the BNT as really kind of the gatekeepers of that activity.
I'd like to talk about that one a little bit. It's certainly part of what the BNT does. The BNT is a land management organization and that works on conservation. But in truth, as part of the government, it's the best commission are the ones that set the policies on having to assess a species need. And that is done also legislatively through the Bahamas government. So it's a push both at best commission as well as to the people that represent you in Parliament.
Yeah, yeah, thank you, Ethan, for summarizing that for us. I completely agree. We do, as the BNT though, and as the managers of national parks and involved, being an involved stakeholder and leader a lot of the times in the protection of both areas and species. It's a lot of yeah, I'd say they are more willing in a lot of the cases to listen to Bahamians as well in some cases. So if you can try garnering support from local communities, and get them involved and teach them why it's important to protect the species. I definitely say to try that approach as well. Another question, I suppose, for Ethan. The sapphire blue hole near Preacher's Cave on Eleuthera is under the threat of increased visitor volume, clearing, etc. This ecosystem seems very rare. Are efforts being made to protect it? I've watched the health of this blue hole decline in recent years.
I don't know of any current efforts to protect it per se. All of that area, I believe, is Spanish Wells commonage land. So which is the same, it's true for Preacher's Cave. So there have been proposals to do protection, but because it was commonage land, at the time, there wasn't a real mechanism to actually take it out of the commonage and put it under a protected status. As far as I know, I could be wrong on that.
Okay. Yeah. Okay. So if I think that kind of wraps it up for the number of questions that we have in the question box. Are there any other questions?
Oh, I just want to correct one last little thing about the blue hole. That is not actually Spanish Wells commonage land. That's Harbor Island commonage land.
Okay, thank you, Ethan, for that clarification. Well, it doesn't seem like we have any other questions. Thank you all for presenting today, for giving your talks. As I said previously, we have 2 more weeks of this series. It's been a great time and an honor to have organized it, as well as being able to learn all of these things about the resources and the species that live in the Bahamas. So thank you again, and I hope you all can join us next week for Into the Deep, exploring the depths of the Bahamas. Thank you, everyone.